a project is about exploring a isoniazid drug efficacy based on a structural perspective. And isoniazid is an anti-tubercular drug, and it is a first line drug used to treat a TB, and it is the cheapest of all the other drugs, and it is available worldwide. So we have took isoniazid as a drug of interest. Our aim is to find the promiscuous drug target within the MTB genome for the particular drug isoniazid, and scope. INHA is a common drug target of a INH, but there are some other drug target which have a similar binding site for a INH NAD adduct, which plays the most important role in activating the MTB genome. Introduction. TB is caused by a Mycobacterium tuberculosis, and it has an unusual waxy coating on its cell surface called mycolic acid. And the INHA is a drug used to treat TB, and it is particularly a prodrug. It is activated by a CATG, a catalase peroxidase enzyme. A mechanism of action. Isoniazid is a prodrug, and it is activated by a CATG, catalase peroxidase enzyme, that are present within a MTB genome. Then the INH bind to the NAD. This NAD comes from a enol acyl carrier protein reductase enzyme, and this INH NAD adduct binds to the INHA, which is nothing but the enol acyl carrier protein reductase, which blocks the mycolic acid synthesis. Methodology. There are 6 steps involved in this process. First is a collection of list of NAD protein, and second is the modeling the protein structure, and third validation of a protein structure, and fourth editing the protein structure, and fifth protein structure comparison, and sixth binding site prediction. In the first step, collection of NAD binding protein. The NAD binding protein are obtained from the tuberculosis database, and with the help of the tuberculosis database, we have obtained the RV number and UniProt ID for the particular protein, and we have collected a 164 proteins. For that, 25 has a structure and 136 doesn't have a structure. Among the 136 NAD binding protein without a structure, ETH protein is mere important because this protein is a target of ethionamide. Ethionamide is a nothing but a drug which is used to treat a TB. It has a same drug target as such of a INH, so it is important to design a ETH protein structure, and the modeling involves 3 steps. First is the template selection, and second is the aligning the protein sequence. The template is chosen by blasting the FASTA sequence against a PDB database, and from that we choose a template, and then we'll align the template to that of a target. After aligning the protein sequence, then the Modeler 9.16 software generates a protein structure. It generates about a 10 protein structure, and among the 10 protein structure, the one with the lowest activation energy, that is the E value, is chosen. Then we have to validate the particular protein structure. The protein structure generated are submitted on a PDB-sum database for a validation, and the result is assessed through an email. And the result should provide the complete detail about the residues that are present in the most favorable region, which is proceeded in a red region, and 90% of the residues should be in that most favorable region, and yellow is the disallowed region, and it should contain only a 2% of a residues. Here this protein contain 86.5% residues in the most favored region and 1.2% residues in a disallowed region, and it is an acceptable value, so this protein can be accepted. The protein structures are edited for the easy comparison. Usually the protein structure have a 4 different chain. Among them only a particular A chain is of important for us, so it is necessary to cut the other 3 chains. So editing the protein structure is also an important step. The protein structure is edited using a Discovery Studio, and the protein structure are downloaded as a PDB file from the PDB database. And this is the example of Fab G1, and we have a complete protein structure of a Fab G1 before editing and after editing. Next is the protein structure comparison. We believe that the protein which are structurally similar also have the same binding site for the binding of INH-NAD adduct. So, it is important to compare the protein structure with the help of a PDB-eFold database. Here, all the other 26 protein with the structure is compared against the INH, and we have grouped into a 3 groups. And here we can clearly see that the Fab G1, Fab G3 and INHA resemble each other about 70%. So we may also say that they can have a same binding site, and also they have a RMSD value less than 2.60. Even though they are structurally not similar, they may have a same binding site. So, binding site comparison is also important in this process. There are 3 important software used for a binding site comparison. They are Ligand Scout and Pocket and Multibind. Ligand Scout is used to find the 3D pharmacophore model, and the Ligand Scout is mainly done to understand the pharmacophore of active sites, and Pocket V is another software that is it compares the complementarity between the binding site and the ligand molecules, and it is also important to visualize the pocket of the ligand molecules. This is a graph which compares the data obtained from the Ligand Scout and a Pocket. The blue lines are the data obtained from the Ligand Scout, and the red is from the, and green is a similarity between these two software. For all the 26 protein structure, we have compared the data from Ligand Scout and the Pocket. Multibyte is an important software which compares the INHA against the other 26 protein structure, and it is an important computational method, and we can easily search the protein structure with the help of their PDB IDs, or else we can upload the PDB file for one protein against the other. Finally, the results. For about 136 proteins which have a NAD as a cofactor are retrieved, and these proteins then searched against a PDB database for the availability of a 3D structure in complex with the NAD. And there is a table shown below which contains a NAD or a NDP or NAI or a NAP as a cofactor. And these are the list of NAD binding protein, and these are the PDB IDs. There are 25 proteins with a structure, and while assessing the PDB ID, we can download their protein structures. And for the protein without a structure, we have modeled a protein ETH because of its importance, and this is the protein structure of a ETH, and it is validated by a Ramachandran plot. And this is a result we have obtained from a PDB sum, and it is obtained as a plot with the 3 different region, the most favorable region with the 89% and the disallowed region with the 2%. Hence it is a good model, and the model is proposed to be a good one. Then, while we have compared the protein structure, we have grouped it into a 3 different group. For a group 1, Fab G1, Fab G3 and INH same structure, and for a group 2, MHPF and AST has the same structure, and for a group 3, Y and SCRA have a same structure. And this is the superimposed image of all three different groups. In the first image, the violet color is a Fab G3 and yellow is Fab G1 and red is INHA. And in the second image, green is a AHCY and violet is a SCAR. And in the third image, blue is a AST and pink is MHPF. Then we have come to a fact that even though the protein structure are not similar, they may have a same binding site. This is because a difference in their folding pattern. So it is important to check the binding site for the binding of INH NAD adduct. The and the binding site comparison is made with the help of multi-bind software. From the multi-bind, we have obtained the following result. In the first 5 proteins, Fab G3, Fab G1, QOR, ALD, AROE have the 50% similar binding site to that of a INHA, while other has less than 50%. And this is the superimposed image of the protein structure when compared with the INHA. Fab G3 and INHA almost have a same binding site, and ROE and INHA have about more than 50% similar binding site, and Fab G1 and INHA have the similar binding site at about a 70%. And this is a QOR and ALD. Conclusion. From the binding site prediction, we have found that almost 26 proteins have a same binding site to that of a INHA except FPRA protein. So this predicts that INHA have a off target which might affect the efficacy of a INHA drug. So we need to improve the efficacy of a INHA drug with the help of the data collected from this